So watch this after we finish talking about the first part of the nervous system, or our first lecture on the nervous system when we finish it in class. This lecture is specifically going to elaborate on what we talk about then, and it is going to cover neurotransmitters, receptors for neurotransmitters, and neuroendocrines, and more about excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So as we discussed at the very beginning of the nervous system, a little bit of a review here. What is a synapse? A synapse is the functional connection between a neuron and the cell that it is signaling. So it's a functional connection between the neuron and another cell. In the central nervous system, this second cell will be another neuron. If you think about an interneuron in a reflex arc, that interneuron synapses with an efferent neuron. In the peripheral nervous system, the second cell will be a muscle or gland. Think of it, in this case, think of that second cell as being an efferent neuron, which is going to synapse with a muscle or gland. These are often called myoneural or neuromuscular junctions. When the neuron in the peripheral nervous system synapses with a muscle or gland. We often think of neurons with regards to synapses as the neuron before the synapse is the presynaptic neuron. The neuron that is immediately after the synapse, or the neuron that's entering the synapse, is the presynaptic neuron. And the neuron that forms the second part of the synapse is a postsynaptic neuron. So think of the synapse as being like a gap. Presynaptic neuron before the gap, postsynaptic neuron after the gap. So presynaptic neuron, the axon terminal of the presynaptic neuron with its axon of the presynaptic neuron, and the postsynaptic cell, whether it be a neuron, a muscle, or a gland. The presynaptic neuron can synapse with a dendrite, a cell body, or an axon of a second neuron. I remember I like to think of synapses as being a snap on a shirt, where there's one side, which would be your presynaptic neuron, and the other side is your postsynaptic neuron. So your presynaptic neuron can synapse with a dendrite, making an axodendritic synapse. It can synapse with the cell body or the soma. Remember, that's what we often call the somas, the cell bodies of neurons. And if you remember that from your Biology 222 class, soma, axosomatic synapse, or it can synapse, the presynaptic neuron can synapse with with the axon of this postsynaptic neuron. So think of this presynaptic neuron, postsynaptic neuron. And I think I'm going to change this in my original PowerPoint. Postsynaptic neuron. Most synapses are axodendritic. So presynaptic neuron is going to synapse with a dendrite of the postsynaptic neuron and are one direction. Synapses can be electrical or chemical. Chemical synapses involve the release of a chemical at the axon terminal. So the release of, or the exocytosis of synaptic vesicles full of neurotransmitter are chemical synapses. Release that neurotransmitter into the synaptic cleft, and that neurotransmitter binds to receptors on the postsynaptic membrane. Electrical synapses are a little different in that the presynaptic membrane of the neuron and the postsynaptic membrane are connected by gap junctions. Remember from preliminary cell biology lectures we've had, gap junctions are tunnels that connect one cell with the next cell, or channels that connect one cell with the next cell and allow ions to move through. You think about the intercalated discs of cardiac muscle. They also are connected by gap junctions. So we can have electrical synapses that connect a presynaptic neuron with its postsynaptic cell, whether it be a neuron, a muscle, or a gland. Most electrical synapses are found in synapses where the postsynaptic membrane is smooth muscle or cardiac muscle. Also, we find some electrical synapses between neurons of the brain and between glial cells. Remember, glial cells are the supporting cells of the nervous system. Thinking specifically about chemical synapses and the release of the neurotransmitter. I'm going to go step by step through the release of a neurotransmitter. First, an action potential, and we're going to talk a lot more, but in the first part, when in class, we're going to talk specifically about an action potential. But the first thing that happens to release the neurotransmitter at a chemical synapse is an action potential comes down to the axon terminal, depolarizing the axon terminal. The depolarization of the membrane on the axon terminal opens voltage-gated calcium channels, and calcium enters the cell. So we have a much higher level of calcium on the outside of the cell and a lower amount inside the cell. And so if we open the voltage-gated channels, which open because of the change in membrane potential involved in depolarization of the membrane, calcium is going to flow into the cell. Calcium entry triggers exocytosis, binds to the membranes of the synaptic vesicles, and triggers exocytosis of the synaptic vesicle contents. Well, I'm going to stop here just for a quick minute because I know my cat is doing crazy things in my kitchen and I need to go see what he's doing. Bear with me for 1 second, okay? What are you doing, bud? Is that the same one you've been playing with the whole time? Sorry about that, I'm back. So, calcium binds to the membranes of the synaptic vesicles, and the synaptic vesicles then have receptors on them that bind to a snare complex, receptors on the surface of the inner surface of the presynaptic neurons axon terminal. And when they bind, that triggers the release of the neurotransmitter. So the neurotransmitter then diffuses across the synaptic cleft and binds with receptors on the postsynaptic cell. So an action let me just review here. Action potential depolarizes the axon terminal. Membrane voltage, membrane potential change along the membrane down to the axon terminal. In the axon terminal, depolarization is going to open voltage-gated calcium channels, and calcium is going to enter the cell. Calcium entry triggers the exocytosis of synaptic vesicle contents because calcium binds to the membrane of the synaptic vesicles, which results in the synaptic vesicles receptors that it already has binding to the receptors on the inner surface of the presynaptic neuron that has receptors for the proteins on the presynaptic neuron. Let me just reiterate, let me just clarify this really quickly. I've been saying receptors on the synaptic membrane. Let's call them proteins. So there are proteins on the synaptic vesicle membrane that bind to these docking proteins or receptors called snare receptors. When they bind, that triggers release of the neurotransmitter into the synaptic cleft. So calcium triggers exocytosis by allowing the proteins on the synaptic membranes to bind to their complementary snare complex receptors on the inner surface of the presynaptic neuron. This triggers exocytosis of the neurotransmitter into the synaptic cleft. These receptors are almost always, actually I'm not going to say almost always, I'm going to say always, a receptor is going to open. So these are ligand-gated channels. The ligand is the neurotransmitter that binds to a receptor on a channel, and there's a channel right here, right next to it. That can then open the channel. So the neurotransmitter is the ligand that's going to bind to a receptor on a ligand-gated channel. Which is going to open that channel and allow a given ion to move in or out of the cell. Talked a little bit in the last lecture that we're going to have in class about graded potentials. When ligand-gated ion channels open, so remember this is when neurotransmitters bind to a receptor that's connected to an ion channel and allow that channel to open, the membrane potential changes depending on which ion channel is open. The opening of sodium or calcium channels results in a graded depolarization called an excitatory postsynaptic potential. So if the neurotransmitter that's released into the synaptic cleft is the ligand necessary to open sodium or calcium channels, that flow of sodium or calcium into the postsynaptic cell is going to result in a graded depolarization called an excitatory postsynaptic potential. Excitatory postsynaptic potential meaning that the membrane potential gets closer to the threshold of where an action potential can be triggered. So the membrane potential becomes more positive, moving more towards the threshold that can trigger an action, potentially trigger an action potential. However, if the neurotransmitter is the right ligand to bind to ion channels for potassium or chloride, potassium and chloride channels will open, resulting in a graded hyperpolarization. Called an inhibitory postsynaptic potential. It's inhibitory because hyperpolarization, remember, we make the membrane more make it, if we hyperpolarize the membrane, we make it more negative, moving it further away from the threshold. Which is going to make reaching threshold less likely. And remember, the reason we have chloride here, chloride follows sodium. It counterbalances the charge of sodium out in the extracellular fluid. So if it flows in, it's going to make the inside of the cell more negative than it already is. Potassium, of course, would flow back out, making the inside of the cell still more negative than it already is on the postsynaptic cell. So opening potassium or chloride channels is going to result in a graded hyperpolarization called an inhibitory postsynaptic potential, because it's a potential change of the postsynaptic cell that can inhibit the possibility of reaching threshold to trigger an action potential, in order to trigger an action potential. So excitatory postsynaptic potentials move the membrane potential closer to threshold, where we can trigger an action potential, make it more positive. Because these excitatory postsynaptic potentials are graded, and only remember they drop off with distance, it may require multiple excitatory postsynaptic potentials from several presynaptic neurons to actually produce an action potential. Inhibitory postsynaptic potentials move the membrane potential farther from threshold, so making it more negative. Inhibitory postsynaptic potentials can counter excitatory postsynaptic potentials from other neurons, so that summation of excitatory postsynaptic potentials and inhibitory postsynaptic potentials, so a positive with a minus, at the initial segment of the axon, next to the axon hillock, determines whether an action potential occurs. So how many positives we have plus how many negatives we have. Do we have enough of a positive change overall that we've reached threshold or not? So we can have multiple excitatory postsynaptic potentials and multiple inhibitory postsynaptic potentials that all work on the same cell body and all work to determine whether or not an action potential is going to occur at the axon hillock. So it's a little bit of a summary of neurotransmitter action. In the presynaptic neuron, in the axon terminal, action potentials move down the axon, opens voltage-gated calcium channels, and there's a release of a neurotransmitter. whether it's excitatory or inhibitory. Dendrites and cell bodies open chemically ligand-gated channels. We can either open channels that are going to result in the entry of chloride, sodium, or calcium, or we can open chemically gated channels, ligand-gated channels, that allow for potassium to be released. In this specific example, with an excitatory neurotransmitter, the neurotransmitter is going to bind to chemically ligand-gated channels that allow for the diffusion of sodium into the postsynaptic cell, resulting in a depolarization. Remember, this is what we call an excitatory postsynaptic potential. The result is that we get a localized decremental. Remember, decremental meaning something drops off with distance. Kind of like waves in an ocean, it drops off with distance. Decremental conduction of an excitatory postsynaptic potential, which as a result opens more voltage-gated sodium channels and then potassium channels. Threshold, the membrane potential becomes positive enough that we can reach threshold and trigger an action potential in the axon of the postsynaptic neuron. Remember that with this postsynaptic cell does not always have to be a neuron. It can be a neuron, but it can also be a muscle. It can also be a gland. So a response to a neurotransmitter can result in a couple different processes. The neurotransmitter can bind to a chemically gated ion channel. Remember, it's a receptor for a ligand, chemically gated ion channel, and allows ion channels to open with sodium coming in to the cell, causing an excitatory postsynaptic potential or excitatory depolarization. Or we can have the ion channels open, and those channels can be channels for potassium or chloride, resulting in an inhibitory postsynaptic potential or inhibitory hyperpolarization, making the membrane potential more negative. This making it more positive so it's closer to reaching threshold. This making it more negative so it's farther away from reaching threshold. We can also have neurotransmitters that bind to G protein coupled receptors. G coupled protein receptors get activated, and as a result of a protein that gets activated, we can alter the open state of ion channels, where some ion channels will close, some ion channels will open, or this can activate a second messenger pathway that modifies existing proteins or regulates the synthesis of new proteins. And there's a coordinated intracellular response. So if the G protein, if a neurotransmitter binds to a G protein coupled receptor, we activate a second messenger pathway that alters the state of ion channels. Either the ion channels can be opened or they can be closed. If they're open, we let more sodium in, resulting in excitatory postsynaptic potential. Or we open potassium or chloride channels, resulting in an inhibitory postsynaptic potential or an inhibitory hyperpolarization. Or alternatively, those ion channels this can result in closure of ion channels, allowing less sodium in, which would be the same as an inhibitory postsynaptic potential, because if we let less sodium in, we're less likely to depolarize the cell or reduce depolarization of the cell. Or we can let less potassium out, resulting in the cell becoming less, the membrane potential becoming less negative, resulting in more of an excitatory postsynaptic potential or an excitatory depolarization of the membrane. Or this can modify existing proteins or regulate the synthesis of new proteins by altering activities within the nucleus, resulting in a coordinated intracellular response. Epinephrine, adrenergic, back here, or to excite postganglionic neurons. This is specifically muscarinic inhibiting cardiac muscle, so slowing the heart rate, inhibiting the cardiac muscle contraction. Atrioventricular node, specialized conducting tissues, slowing conduction velocity. Vasodilation of smooth muscle, so excitatory. Vasodilation of heart, brain, and external genitalia, so excitatory. Excitatory vasodilation. Sympathetic varicosities, inhibiting norepinephrine release. With regards to norepinephrine, adrenergic receptors, remember we have alpha and beta receptors. Alpha receptors work through phospholipase C to increase mucus in saliva, constrict arteries, constrict bronchioles, vasoconstriction. So performs these functions through vasoconstriction. Alpha 2 receptors decrease cyclic AMP, inhibiting digestive processes and inhibiting insulin release. So specifically an inhibitory response. Beta 1 receptors, specifically impacting the heart. Excitatory, increasing the heart rate, increasing contraction force, and increasing autorhythmicity. We're going to call it that, autorhythmicity, so increasing the rhythm that the heart contracts. Remember how the sinoatrial node initiates this. Don't know if you learned that in Biology 222 or not, but the sinoatrial node is going to be stimulated here to increase rhythm of heart rate. Beta 2 receptors result in relaxation of smooth muscle. Beta 3 receptors stimulate lipolysis, or breakdown of lipids. Remember, lysis always means the breakdown. So beta 1 receptors work through decreasing cyclic AMP to dilate veins, inhibit digestive processes, and dilate bronchioles. It's going to activate things. I completely read that wrong. Beta 1 specifically increasing heart rate. Beta 2 inhibitory. Through decreasing cyclic AMP, dilates veins, inhibits digestive processes, and dilates bronchioles. Beta 3 receptors increase cyclic AMP, resulting in increased fat breakdown or lipolysis. Talked a little bit about this, and you'll see it when we in class. The graded nature of excitatory postsynaptic potentials excitatory postsynaptic potentials can sum with other excited postsynaptic potentials, and also can sum with inhibitory postsynaptic potentials. But when multiple excitatory postsynaptic potentials sum, they have the possibility of reaching enough of a positive membrane potential, going up from -70 to -50, to where threshold is reached and an action potential is initiated. So we can have the summation of multiple excitatory postsynaptic potentials, or we can have the summation of excitatory postsynaptic potentials with inhibitory postsynaptic potentials. They can sum in a temporal summation, which is a timing. Temporal, think of temporal as temporal existence, temporal time. Temporal is meaning a timing. How quickly did the one synaptic potential come in and the next synaptic potential come in? coming from the same postsynaptic neuron, how quickly are they initiated, the action potentials? And when they come in, do they come in quick enough that the membrane has not completely reached the resting membrane potential, and the next one just adds to the remaining membrane potential change that has already occurred? It's temporal summation. Also have spatial summation, where multiple postsynaptic neurons are acting on one multiple presynaptic neurons are acting on one postsynaptic neuron. And their summed synaptic potentials results in a reaching of threshold and the firing of an action potential. Or we can have multiple presynaptic neurons synapsing on a postsynaptic neuron, resulting in the spatial summation of either excitatory postsynaptic potentials together or excitatory postsynaptic potentials with an inhibitory postsynaptic potential, or multiple excitatory postsynaptic potentials and multiple inhibitory postsynaptic potentials summing together, and the final result of the membrane potential not being able to reach threshold and fire an action potential. So as a comparison of excitatory postsynaptic potentials, remember they cause depolarization, action potentials cause depolarization. Action potentials, bear with me, action potentials are always depolarizing. An excitatory postsynaptic potential with the depolarizing with the result of sodium gates opening. Excitatory postsynaptic potentials are where acetylcholine or other neurotransmitters bind to ligand-gated channels, resulting in the opening of sodium or potassium gates, or the closing of, sodium or calcium gates, or the closing of potassium gates and the opening of sodium gates. Action potentials, the initial effect is always the opening of sodium gates. Cause of repolarization in the case of an action potential is always the opening of potassium gates. Excitatory postsynaptic potentials, remember that these are decremental, so they drop off with time and distance. The loss of intracellular positive charges with time and distance. Conduction distance in the case of an action potential is because it's regenerated over the length of the entire axon. And excitatory postsynaptic drops off with distance, and it's a localized potential. With an action potential, there's positive feedback between depolarization and the opening of more voltage-gated sodium channels. In case of an excitatory postsynaptic potential, that isn't the case. Maximum depolarization for an action potential is about positive 40 millivolts. For an excitatory postsynaptic potential, it's close to 0. In the case of an action potential, there's no summation. It's an all or nothing event. Regardless of how strong the stimulus was of the postsynaptic potential or how strong the firing was, it either occurs completely or it doesn't, and a higher stimulus will not result in a higher magnitude of an action potential. In the case of an excitatory postsynaptic potential, there can be a summation of synaptic potentials resulting in graded depolarizations. In the case of action potentials, there is a refractory period. And remember, we have the absolute refractory periods and relative refractory periods. In the case of the excitatory postsynaptic potentials, there are no refractory periods. In the case of the effects of drugs, acetylcholine effects that are stimulated by action potentials can be inhibited by tetrodotoxin, but not by curare. Acetylcholine effects in excitatory postsynaptic potentials are affected by curare, but not by tetrodotoxin. Last part of this chapter, about convergence versus divergent pathways. A divergent pathway is where one presynaptic neuron branches to affect a larger number of postsynaptic neurons. So when a signal diverges, the signal from one presynaptic neuron diverges to affect a larger number of postsynaptic neurons. In a convergent pathway, many presynaptic neurons can converge on a smaller number of postsynaptic neurons. This is where we get the summation, think about that, of excitatory and inhibitory postsynaptic potentials. When many presynaptic neurons can converge onto a smaller number of postsynaptic neurons. Last slide. We can have presynaptic inhibition and postsynaptic inhibition. Presynaptic inhibition is going to occur before